So, good evening, everyone. In the online expert talk of ISFCP, our today guest is the Dr. Ashok Kumar Jaishankar. Uh, Under the aegis of this talk, we are arranging under the aegis of the IQAC ISFCP. I am having privilege to introduce Dr. Ashok Kumar Janakiraman. He is an associate professor in the Faculty of the Pharmaceutical Science at UCSI University, Malaysia, which is a rank 218 in the QS World University ranking of the 2023. His research is situated in the field of the pharmaceutical technology and product development with a special focus on the nanotechnology for the drug solubility enhancement, fast melting tablet, topical product, and collaborative R&D in the industries such as the pharmaceutical and the cosmeceutical industries. He is one of the brilliant personality and author and co-author of more than 15 research publications, more than 25 conference contributions, 4 patent, 1 trademark, 2 book chapter. He has received several important recognition for his research career, including the Asian Young Innovation Award, silver medal in the Malaysia Technology Expo 2020, the Gold Award in the PECI PTA in 2022 International Conference, and the Exposition on Innovation by the Institution of Higher Learning in Malaysia. So, with this wide range of expertise, today we are having the Dr. Ashok Kumar Janakiraman ji. So, I am inviting Dr. Ashok Kumar ji, please share your view on the said topic of zinc oxide nanoparticles, cutting edge studies, and its biomedical application. Dr. Ashok, over to you.
Thank you, Prof. Nitin Sharma, for the wonderful introduction. And good evening to everyone. This is Dr. Ashok. I'm from UCSI University. So I want to take this opportunity to thank IFS Moga management and director Prof. Gupta ji and today's moderator Prof. Nitin Sharma for providing such opportunity to share my views on the zinc oxide nanoparticle. So I think without further delay, maybe we can start our session. Now I am sharing the slides from my side. Prof. Nitin, can you see my slides here?
Yes, all right, sir. This is visible.
Now it's in full view. Maybe I will make it magnify it. Is it okay? Shall I start now?
Yes, sir. Please go ahead.
Okay. So for the today's online expert talk, we will I chosen the topic of zinc oxide nanoparticles cutting edge studies and its biomedical application. Actually, this study I am doing already in my lab with my lab mates as well as the collaborator from the other university. So maybe I can share a few things because this is a wide topic, but we cannot go in depth. But the allotted time is for me is 30 minutes, so I can able to cover the maximum as much as possible during this 30 minutes time. So in this topic, firstly we will go and have a overview of zinc oxide nanoparticles and what are the synthesis method or what are the methods we are using to prepare the zinc oxide and what are the characterization technique, and especially we are focusing here on the biomedical application. So we can classify this biomedical application by nano carrier or as a drug for many users. Little bit introduction about the nanoparticles. So Richard Feynman is the first man who proposed the concept of nanoscience in 1956. Amongst all the nanomaterials, the nanoparticles have been received as a great deal of attention due to their small size, large surface area, the properties which provide the researchers with a novel way of diagnosis and treating the diseases. The nanoparticle with the diameter ranging from 10 nanometer to 1000 nanometer, but we will preferably climb that nanoparticle is range from 10 to 200 nanometer is considered as a complete nanoparticles. So which therapeutical bioactives to interest or either by encapsulation or conjugated or absorbed onto the surface. We can claim ours is a nanoparticle. The bioactive material it may be encapsulated or absorbed on the surface, whether either in a encapsulated or in a absorbed on the surface. So we can claim in the two ways. We talk about the metal oxide nanoparticles. Now it's grown into the valuable material for the drug and health industries because of their multiple advantages. We can say here is actually it's in a favorable size, the negative surface charges of nanoparticle, especially for the absorption and the elimination of a reticuloendothelial system. This is the main drawback with the nanoparticle. It can be easily eliminated through reticuloendothelial system, but the metal oxide can escape from this absorption and elimination from the reticuloendothelial system. This is the added advantages of a metal oxide nanoparticles. The metal oxide nanoparticles can interact on the more depth in the level and various cellular structures compared to their bulk counterparts, meaning that if you have any precursor, as compared to the precursors, metal precursors, the nanoparticles will have the in-depth level. It can be easily penetrate to the cell membranes. And also they do not cause any systemic toxicity due to their high improved bioavailability. These are the added advantages of metal oxide nano particles. So we have different types of the metal oxide nanoparticles. It starts from cadmium, cerium, iron, magnesium, nickel, silver, titanium, zinc oxide, and uh, zirconium oxide. In that, we are choosing today's talk more focus on the zinc oxide nanoparticles. So we have a different method for the preparation of zinc oxide nanoparticles. We can broadly classify the synthesis method into top-down and bottom-up. Top-down is nothing but the breakdown of large molecule to generate the required nanostructure from them. So it's maybe we can say that it's breakdown of large molecule into small molecule.
Dr.
The
Saab, I
second one,
think I think your
yeah,
slide is not moving, sir.
is it?
Your slide is not moving. Are you still on the first slide?
No, I'm in the fourth slide now.
But your first slide, there is a visualization of your first slide only.
Is it 1 minute? I can reshare
Yes,
again.
right now. This is the synthesis of zinc oxide nanoparticle. Na?
Yes, this is a synthesis of zinc oxide nanoparticle now.
Sir, please reshare your slide.
Yeah, I'm doing now. So now you can able to see the synthesis of zinc oxide nanoparticle slide
Yes, this
there
is
on
the
the screen.
synthesis of zinc oxide nanoparticle.
Yes, now I make it a full screen. Yeah. So, so we have two different types of method for a synthesis of zinc oxide nanoparticles or metal nanoparticles. The first one called as a top-down technique. It is nothing but the breakdown of large molecule to generate the required nanostructure from them. That means breakdown of large molecule into the small molecule. The bottom-up techniques, we can say this is a assembling of a single atom or molecules into the large molecules. So the top-down again classify into milling, laser ablation, uh, sputtering, grinding techniques. The bottom-up we have a salt to gel. Mostly the industry prefer to uh, go with the salt to gel synthesis technique to produce metal oxide nanoparticles. Then the next one is a green synthesis. So the green synthesis is now is most popular technique to prepare the the metal nano oxide. Here is a zinc oxide nanoparticles. We can prepare by using the microorganism, plants, or biological, biological materials. Then pyrolysis is also a laser pyrolysis is also a method to prepare a zinc oxide nanoparticle, but mostly they were used in the industries. For the large scale, they are using the laser pyrolysis method. There is nothing but decomposition uh, brought about by using the temperature. By using the temperature, we can break down or decomposition of materials. So just I am showing here the picturization of each methods. I am not going to talk much on this synthesis. So this is the overview of synthesis of.
Dr. Ashok, I think again your slide is become stuck, sir. You can do one thing, key, sir. You can change the slide manually, sir.
Yeah, okay. Maybe with the full screen will not work, right? I think now you can see
Yeah, it's
the
changed, sir.
slide. Okay.
Please follow this only. Yes,
Okay,
yes.
okay. So we will talk about the zinc oxide nanoparticle. Normally, it's found on Earth crust as a mineral zincite, but not very rare to get naturally the zinc oxide. Usually, we will produce synthetically. It's a white, colorless crystalline powder, and it's have a biocompatible characteristic, and it's a very good semiconductor. The molecular weight around 81.38, and it's safe by already approved by USFDA as a safe active ingredient or excipient. We can use it for a drug or if any medicinal uses. So this is the overall information about the zinc oxide nanoparticles. So after preparing zinc nanoparticles, we need to do the characterization. So we have a lot of characterization technique to confirm the zinc oxide and the drug which is already encapsulated or it's absorbed on the surface. The first and foremost analysis is a particle size and zeta potential. So we need to check the particle size of prepared zinc oxide nanoparticle by using the zeta sizer. Then the morphology we can confirm through the SEM or TEM analysis or EDX, energy dispersive X-ray analysis. We can have a coupled TEM and EDX that is used to the chemical composition and mapping of zinc oxide. We can do individually or we have to merge both TEM with the EDX. And the XRD, of course, this is actually to confirm the crystallinity of nanoparticles. So the crystallinity of nanoparticle has been confirmed by using the X-ray diffraction, and for the molecular groups and chemical bonding, of course, by using the FTIR, and synthesis of mechanism we will confirm by the thermogravimetry analysis or differential thermal analysis. Then the band gap absorption peak by using the UV spectroscopy. And nowadays, for the surface functional nanoparticle, they will use the X-ray probe electron spectroscopy as an instrument to confirm the surface properties of a prepared material. That is called as the X-ray photoelectron spectroscopy. So this is some of the pictures which is related to the characteristics of the zinc oxide nanoparticles. We have a UV FTIR to confirm the uh, the molecular bands. Then for the application of zinc oxide nanoparticle, we can say zinc oxide is used almost all the fields, including agriculture, rubber, cosmetic, uh, paints, industries, then textiles, and the biomedical as well. So our the talk is mainly focused on the biochemical, sorry, biomedical. So in that biomedical, we will have broadly we can classify into two: the nanocarriers and drugs. The drugs may be the anti-cancer, antibacterial, antifungal, orthopedic implants or bone healings, anti-inflammatory, anti-diabetic, anti-accident, antiviral, and cardioprotective action. Maybe we can say that the zinc oxide may be used as a drug for many diseases. The first one is a nanocarrier. As you know, the nanocarrier is used to transport the substances, either if it is a drug molecule or if any biological thingy, then we can use it, the zinc oxide, as a carrier to transport the substances. So you look at the picture here. Is firstly, it's used as a surface functionalization of a zinc oxide nanoparticle for active drug delivery to the cancer stem cells. If you look at the picture, so we have the prepared zinc oxide nanoparticle. It have interact with the, or it have attached with the cancer stem cells or with the tumor cells. Then it having the surface functionalization here. Okay, that's maybe we can call it as this is a cancer cell marker, so it can attach to the cancer cell, not to the normal cells. So once it's get attached, then it's entered through the endocytosis mechanism. If you look at the second picture, the prepared zinc oxide nanoparticle will enter into the cell. Then it releases the zinc oxide from the prepared zinc oxide nanoparticle. So it's a dissociation of zinc molecule from the nanoparticles. And also we can incorporate drug along the main inside the zinc. Then once the drug get released into the site, then it may produce a action. So maybe we can say it can be kill the cancer cells. Then the third one, if you look at this picture, there is a stimuli response zinc oxide nanoparticle mediated drug release to the cancer stem cells. So either by external stimuli or internal stimuli, we will use the external or internal stimuli to release the drug through zinc oxide nanoparticles. So external stimuli like heat, ultrasounds or light sources, we can use to release the drug into the targeted place. The external stimuli like pH, redox and enzyme activity also can use to release the drug to the targeted site. So here the zinc oxide role is considered or act as a carrier to transport the drug from the administered place into the targeted place. So let's say we can nowadays in the advanced zinc oxide, functionalized zinc oxide nanoparticles, they may be having the surface coating. So the surface coating with a drug or if any polymer or biopolymers or inorganic moieties, materials or bioconjunction with the any functional specific functional groups that can be used to be a theranostic agent. Okay, not only to treat, it also used to detect the cancerous cells. So we can call it as the functional multifunctional zinc oxide nanoparticles is used as a theranostic agent to diagnosis as well as treat cancers. So some of the examples for zinc oxide nanoparticles, which already published uh, to the reputed journals. So zinc oxide nanoparticles with the doxorubicin, they checked against the lungs cancer cells uh, to ensure the activity. So it's already having activity. That means to kill the cancer cell line. So they proven already in vitro. So likewise, we have zinc oxide nanoparticle with a polyethylene glycol, zinc oxide nanoparticle with chitosan, and zinc oxide nanoparticle with the amine. Then we have a chitosan zinc oxide nanoparticle, diallyl hydrate cellulose hydrogels. This may be for a topical application. And also we will, they already proven that the zinc oxide nanoparticles will be antibacterial activity. So checked against the Streptococcus aureus, and it have mm, compared with the standard drugs or controls. Then the zinc oxide may be act as a anti-cancer agent. So the main mechanism is endocytosis. You know that the endocytosis is nothing but the substance are brought into the cell. So by engulfing, it enter into the cell. So this cell is considered as a assume this is a cancerous cell, the zinc oxide nanoparticle will already enter into the cells through the endocytosis mechanism. Then once it's get enter, it releases the zinc ions. So you look at the picture, we have 4, 5 zinc ions inside the cell. Then once it's get released or enter into the cell, then it produce the ROS production. That is, uh, this is a oxygen species, reactive oxygen species production will start. Once it gets start, it may be produce the oxidative stress, or it may damage the DNA molecule which is present inside the cancer cells. Then the reactive oxygen species production may be used to damage the DNA cells through intrinsic apoptosis and extrinsic apoptosis. So the intrinsic apoptosis means it's inside the cell. The extrinsic apoptosis means is on the surface of a cell wall. So either uh, through this ROS, oxidative stress, or DNA damage, or the uh, destruction of the lipid or protein in the cell, it can be able to kill the cancerous cell. So the another mechanism of the cytotoxicity potential of the zinc oxide nanoparticles, either by the pro-inflammatory responses. Okay, the pro-inflammatory cytokines can be produced once the zinc oxide nanoparticle enter into the body. So either by the pro-inflammatory response, or the redox reaction system, or sometimes directly metal ions, the zinc mediated uh, desiccation process of a protein activity. So straight away that can be act. Here also the mechanism behind that is either through the oxidative stress or direct DNA damage. So the cancerous cell cytotoxicity of cancerous cells we can observe, or we can get after administrating the zinc oxide nanoparticle. Either one of this mechanism, or sometimes it may all together, all three mechanism will work and kill the zinc oxide nanoparticles. So what is the other advantages as compared to the normal nanoparticles? This mostly used for the if sometimes the drug can have a resist against the particular cancer cell. So in this case, we can use the zinc oxide nanoparticle. Then the potency and the efficacy wise, as compared to the normal nanoparticle, the zinc oxide will have proven to have a higher efficacy, especially in the case of drug resistant on the cancer cells. Then this is the antibacterial activity of a zinc oxide nanoparticle. So again, it's having the three mechanism. The first one is a dissolution and the ion release. The second one is a reactive oxygen species generation. So once it's get reactive oxygen species, then the membrane dysfunction. So it is then the nanoparticle internalization into the cell. So you look at the picture, we have uh, the first step is a ROS production. Second step is the release of uh, zinc oxide or zinc metal ion. The third one is encapsulation. That means the endocytosis. So enter into the cell. The other side, the right side, we have a picture. So this is our zinc oxide nanoparticle. It may be normally we can measure the antibacterial activity through zone of inhibition, right? So if the zone of inhibition, we can measure and compare with the control drug or the standard drug. Then the next one picture, we have the fluorescence microscopy. So this is for applying that means the live cells before administration of a zinc oxide. So the red one, it's indicate that already the cancerous cell or live cell has been destroyed. After this one, the B one is after administration of the zinc oxide nanoparticles. Then the next concept is a skin treatment. So we have already some proven concept for a skin treatment. Zinc oxide nanoparticle, or sometimes zinc oxide nanoparticle loaded with the chitosan. So basically it can be promote the wound closures, and also it used to promote the reepithelialization and the collagen formation on the surface. So it restore the skin as a normal normal condition. So you look at the picture A and B. So the A is having the live bacteria, that means the wounded skin, as compared to the B picture or figure. You can see we cannot find any live bacteria here. So only the dead bacteria. So it has proven already it's having the wound healing activity. Then the next one is the anti-inflammatory action for zinc oxide nanoparticles. It's actually to inhibit the release of pro-inflammatory cytokine interleukins, and also the immune system activation. So it blocks the pro-inflammatory cytokines and to produce a immune action. That means it has been activate the immune system in our body to prevent anti-inflammation. This is the mechanism. So we will have acute inflammation through the master cell activation that can be and overcome, or that can be inhibited by using the zinc oxide nanoparticles. And we can say this also used for orthopedic implants on the bone, uh, bone replacement or whatever it may be. So it's bone related operation or any surgeries, then we can use as a zinc coated material. So you look at the picture. So we will have the zinc coating materials. uh, So it may be used to prevent the bacterial growth on the surface, that means on the joint place. And also it may be produce the growth factors like IGF-1 or IGF-1 or IGF-2 to enhance the growth of bones. So this is actually the zinc oxide have the versatile use as compared to other nanoparticles, the metallic nanoparticles. So if you look at all the advantages of zinc oxide, we used to see some disadvantages as well, and also what are the problems or what are the issues currently the researchers are facing while prepare the zinc oxide nanoparticles. Mainly, if it is a favorable size and it should have the negative charge on the surface that can be easily enter into the or easily escape from the absorption uh, or elimination from the reticular endothelial system. If the size is very slow, sorry, very less. For example, your nanoparticle size is below 20 nanometers, then it may be produce the cell cyto toxicity effect. That means the normal cell will get affected because of the particle size. So we cannot be make it the particle size very less. It should have some average range, then the favorable size only can produce the effective treatment. So we look it into this area while we prepare the zinc oxide nanoparticles. And also we need to have an attention to the shape of the particles. So if there are kind of nanospheres and nanosticks, nowadays they are using more drug loading. So suppose they wanted to have a high quantity of drug, want to encapsulate, then in this case they can use the nanosticks and nanospheres instead of nanoparticles. and of course the concentration. We cannot consume much concentration because that may produce a toxicity to our uh, cells or tissues. Then this is my conclusion and future perspective of the metal oxide nanoparticles, especially for the zinc oxide nanoparticle. Due to their small size and unique characteristic, nanoparticle possess different properties as compared to other bulk counterparts that's existing and the ever-growing biomedical application, mainly due to their inherent ability to induce an overproduction of ROS and uh, implicity of cell death. So based on our previous slides, maybe you can have an idea. So it's maybe produce an overproduction of ROS as compared to other nanoparticles. This unique properties turn the metal oxide nanoparticles into the suitable anti-cancer, antibacterial, pro-regenerative tools, either as a um, standalone agent or drug delivery platform for the various bioactive molecules. This is based on their unique nature that may be used as for a various bioactive molecules. Furthermore, it will well-known fact that the toxicological risks of any given substance is determined both by its inherent toxicity and exposure time. As we seen in the previous slide, so the concentration, definitely if the concentration is increased, toxicity should be increased. So we need to be very careful and find out what is the suitable concentration we can administer to treat a particular element. So therefore, the exposure time is kept to minimal threshold, and the toxicity risk must be reduced even through the uh, metal oxide nanoparticle possess a powerful cytotoxic effect. So we need to be very careful to while fix the uh, in vivo studies or for a clinical studies. So we due to this reason only we cannot have a much nanoparticle on the market. So this is the main drawback. So owing to ever-growing importance, the metal oxide nanoparticles in the field of biology, medicine, and incremental needs to risk assessment of the toxic effect of metal oxide nanoparticles should be further studied and characterized. So once this challenge of systemic documentation is addressed and overcome, in the future, metal oxide nanoparticles in the biological field will be hold on a ever greater promise, and not only to multifunctional therapeutic strategies, but also for every diseases diagnosis as a therapeutic platform, theranostic platform for in-depth and non-invasive cellular and tissue imaging. So as per our experience as well, we need to do a lot of studies before going to finalize or scrutinize the formulation and the concentration. So that's all about the overview or the points which is related to the zinc oxide nanoparticles. These are all the reference I took from the published article. And this is our team publication related to zinc oxide nanoparticles from past few years. Currently also we communicated few manuscript. This all under review. Hopefully will get published soon. So thank you for your attention. If any question, then feel free to ask me.
Thank you, Dr. Shokji, for such a wonderful presentation on zinc oxide-based nanoparticle. Thank you very much once again for sharing your view on such a gray area. Basically, as a pharmacist, we are working on the polymeric nanoparticles. Rarely we are focusing or giving the emphasis on the zinc oxide or any metal nanoparticle. But your view, your suggestion, your methodology was excellent and extraordinary. And definitely, since we are having the time constant, but your strengthened mechanism of your presentation definitely will help to the all students or the viewer of this video. We are going to put this video on the YouTube. Whatever the suggestion, whatever the question will be asked by the students, definitely will be in the situation to give the answer after your concern. Thank you very much once again, sir. Now, for the conclusion of the session, I would like to invite our respected director, sir, Dr. G.D. Gupta, sir. Sir, over to you.
So thank you very much, Dr. Nitinji. So first of all, I welcome Dr. Ashok Kumar ji, and I am also thankful for accepting our invitation for this ISF dialogue series. Not only this, I am thankful to the UCSI University, Malaysia, for MOU with the ISF College of Pharmacy. So as we know that this types of the MOU, it will be the very useful for the academia as well as the research, and I hope that coming days we can do also some of the joint project with your university. So strength of each and every university have their own strength, and this strength dependent on the quality of the education research and who is the researcher is there. So I come to know that Dr. Ashok Kumar ji, you are the very dedicatedly doing the work on the nanoparticles. This is the related to the especially for the you can say metals, zinc oxide, and so many other things you are doing there. And ISF College of Pharmacy strength is the pharmaceutics, pharmacology, and we have the strong staff as well as the laboratory in the pharmaceutical chemistry also. So I hope that in the coming days, if you have any types of the need from the India, because we have the strong HR management, strong we have the resources of the researcher over here, and I will discuss with you in the coming days. This MOU will give some of the outcome, and this outcome will be more fruitful and useful to the society, either from the India as well as the Malaysia also. I know that number of the scientists and number of the researcher, they are from India to the Malaysia, and they are doing the excellent job. So for this MOU, we are. I am also thankful, Dr. Nitinji, for their very dedication and their invention with this MOU, and he did a lot of efforts for this one. Even that we want to this MOU in the physical mode at a one platform, either in Malaysia or the ISF College of Pharmacy in India, but due to some reason we are not did on the like a physical mode, so not a problem at all. In the coming days, we can organize some of the big event. This is known as the ICS 2026. So I hope that we can also do some of the joint efforts in India. First time going to the organize. This is the International Cyclodextrin Society Conference. So I hope that Malaysia University also contribute with this International Cyclodextrin Society based of the conference, and first time India is going to organize. This is the 22nd one of the ICS. So thank you very much, sir, for your nice. You have given very scientific, technical talk, and I hope that this lecture will be the useful for the pharmaceutical person and those person who are working in this area. So thank you very much, and from core of my hearts, I welcome and thankful to the Malaysia University. Please convey my regard to all your officials, those who are involved in the MOU. Thank you, sir. Thank you very much.
Thank you,
Thank you,
Prof.
Dr. Ashok.
Thank you, Prof. Definitely, I will convey the things to our dean as well as the top management for your willingness to continue as a in the MOU. Hopefully, very soon, maybe we can arrange the physical meeting to have a MOU signature. I think, if I'm not mistaken, from our side, they are doing the documentation. So hopefully we will get the signature soon. I will convey these things to my dean and the higher officials as well. So I once again thank you for allowing me to have a speech on this topic of zinc oxide nanoparticles. I hope that this talk will give you a much clear understanding about the zinc oxide nanoparticle preparation and characterization technique. Thank you once again. Thank you.
Thank you very much, sir. Thank you very much.